I'm Michihiko Nakano from Kyushu University, Japan. Today I want to talk about DNA detection using microbead-based dielectrophoretic impedance measurement. This is a method to detect the DNA amplified by PCR. This is for the conventional PCR. Using this method, amplified DNA by the conventional PCR is detected within 50 minutes. This slide shows the outline of today's talk. I want to start a brief introduction of the dielectrophoresis and the dielectrophoretic impedance measurement, DEPI. Then I want to show you the aim of this study, why we focused on the detect the DNA amplified by PCR. Then I want to show you a new DNA detection method using the microbead dielectrophoresis. And I will show you the experimental results. Then finally I will conclude my presentation. So let me start my presentation with a brief introduction of dielectrophoresis and the dielectrophoretic impedance measurement. Dielectrophoresis is one of the electrokinetic motion acting on the dielectric particle. When the dielectric particle pressed on the under non-uniform electric field, the DP force acting on the particle. There is two types of DP force. One is positive and negative. DP depends on the polarization of the particle and the medium. When the polarization of the particle is higher than the medium, the particle goes to the high electric field region. This is called as a positive DP P-depth. On the other hand, the polarization of the particle is lower than that of medium. The particle goes to the weaker electric field region. This is a negative dielectrophoretic force. Under the microelectrode configuration, so in the case of positive DP, the particle goes to the edge of microelectrode and breaches the shortest gap of the microelectrode. On the other hand, in the case of negative DP, the particle goes to the particle is repelled from the microelectrode. The polarization depends on the conductivity and the permittivity of the particle and the medium. DP also depends on the conductivity and the permittivity. These videos are the typical example of the DP. This black region shows a microelectrode fabricated on a glass substrate. This is the east cells, this bright spot. When applied AC voltage, east cells goes to the edge of microelectrode and aligned along with the electric field line. This is the dielectrophoretic of adenovirus. Diameter of the adenovirus is 100 nanometer. Virus is also trapped by the positive DP on the edge, on the gap of the microelectrode. And the DP depends on the frequency of the applied voltage. We use the dielectrophoresis to detect the bacteria and the virus by electrical operation. We call it dielectrophoretic impedance measurement and DEPI. This is an image of a DEPI. So we can get the signal of the dielectrophoretically trapped target as the impedance change of the microelectrode. DEPI is electrical operation, and we don't need no chemical reagent. This is a commercially available DEPIM device. This device detects oral bacteria in 1 minute. This table shows DEPIM detection of the bacteria and virus. This is E. coli and the adenovirus. This is the DPIM responses. This is the time. This is the conductance increase. So DPIM response is increased with increasing the concentration of the target. Therefore, DPIM can detect the target quantitatively. So we have studied the electric detection of bacteria and virus. As the next step, we try to detect the DNA, especially for diagnosing the virus infection. So, therefore, we try to detect the DNA molecule amplified by PCR. PCR is a method to amplify DNA molecules exponentially from the target DNA molecule. DNA is a powerful tool for molecular biology and virus infection diagnosis. In diagnosing virus infection, DNA is known as one of the most sensitive and specific detection methods. DNA extracted from the virus is amplified by PCR. PCR amplifies DNA from the specific region of the target DNA. A sensitivity of the DNA typically 10 to 100 viruses in a microliter sample solution. After PCR, amplified DNA should be detected. So this slide shows how to detect the DNA amplified by PCR. In the conventional PCR, amplified DNA is confirmed by a dielectrophoresis. In the dielectrophoresis, the amplified DNA is separated by its molecular weight and stained with the fluorescent dye. This process needs a long time, about 1 to 2 hours, and needs manual operation by experts. On the other hand, there is a real-time PCR, as the previous presentation. Real-time PCR monitors DNA amplification during PCR. Real-time PCR needs no post-PCR process, but apparatus for real-time PCR is very expensive, more than 10 times of the conventional PCR. And to design the fluorescent probe to monitoring the DNA molecules, significant knowledge is required. Therefore, we aimed to develop a rapid and easy electrical DNA detection for a conventional PCR. So I want to show our new method to detect the DNA using the microbeads dielectrophoresis. We use dielectric microbeads to detect the DNA. In the method, amplified DNA is attached onto dielectric beads. Then, these DNA-labeled microbeads are detected by DPIM. This is the flow of the detection. So, DNA amplified by PCR are attached on the microbeads. Then, these DNA-labeled beads are detected by DPIM. So, what is the new point of this method? So, DNA labeling, DNA-labeled microbeads dielectrophoresis is not new. Sorry, I skipped the time. So this, sorry. This labeling completed within the 50 minutes, and this is 1 minute. So I want to show you what the new of this method is. We found the phenomena DNA labeling changes the dielectrophoresis of microbeads. When we choose the appropriate diameter of the microbeads, DNA labeling makes dramatically change of dielectrophoresis. Before DNA labeling, dielectric microbeads show the negative DEP, like this one. After the DNA labeling, DNA-labeled microbeads shows positive DEP. We found this phenomenon, and this realized selective selective detection of the DNA-labeled microbeads. Only the DNA-labeled microbeads are trapped by the positive DP. Okay, I want to explain the theory of the microbead dielectrophoresis. Dielectrophoresis of microbeads depends on the conductivity. And the conductivity of the beads depends on its surface conductance, expressed as this equation. And the surface conductance of the beads is proportional to the surface charge density. Therefore, when the surface charge density increases, the dP of the beads is changed. This is the structure of DNA. DNA molecule has a large amount of negative charges because each of phosphate base has one negative charge. When the DNA molecule attached on the dielectric microbeads, the surface conductance Ks increases. This Ks increasing changes the dielectrophoresis of the microbeads. Okay, I will show you experimental results. So, the amplified DNA were prepared from the HIV RNA. And one end of the amplified DNA has a biotin. So we use the streptavidin modified microbeads, and the diameter of the beads was 2.8 micrometer. This streptavidin specifically binds to the biotin. Using streptavidin-biotin interaction, so only the amplified DNA is attached on the DNA-labeled microbeads. So these are videos of the dielectrophoresis of microbeads. So this is in the case of pristine microbeads. This shows the negative DP. This is the electrode and this is the beads. When after DNA labeling, dielectrophoresis is dramatically changed like this. So microbeads goes to the gap of the microelectrode. So this is dipping response of the DNA-labeled microbeads. So x-axis is the time in second. So y-axis is the conductance increase. As you can see, only DNA-labeled microbeads shows the dipping response because the DNA-labeled microbeads trapped between the edge of microelectrode. On the other hand, pristine beads repels from the microelectrode. So because this is a real-time image, because the dielectrophoresis is very quick, so we can get the signal of the dipping so within the 5 to 10 seconds. Okay, we confirmed that this detection method can detect the range DNA size 150 base pair to 1,500 base pair. Okay, so I want to show you new results so quickly. So in the new method, we cannot know a size of DNA molecules, but we can know the DNA size using a frequency sweep. This is a frequency sweep image for DNA size 200, 400, 800. So this is first, this is a negative first derivative of this curve, and plots the peak, we can find the size of the DNA molecules attached on the microbeads. Okay, conclusion. We developed a new DNA detection method using the microbeads dielectrophoresis. And this method utilizes the DNA dielectrophoretic change induced by DNA labeling. And this method needs a 15 minutes labeling and a 10-second electrical detection. And the future work, we want to detect the DNA quantitatively and the size specifically. And we try to combine this method with chip-based PCR. Thank you for your kind attention.